Thank you. It's a pleasure to be here. I have to say I've really enjoyed this morning's talks, and it's great to have a community to interface with around personalized nutrition. So I'm going to tell you a little bit about the work that we're doing at the Institute for Systems Biology, and in particular, a project that we have initiated recently that's generating lots of data for analysis that we'll go into today. So I'll start with disclosures. As already mentioned, I am the co-founder, along with Lee Hood and Clayton Lewis, of a company called Deriving, which I'll mention a little bit because they help fund the 100-person wellness project that I'll describe. I'm also an advisor to Habit, which is a personalized nutrition company. Okay. So as will not be a surprise to anyone in this room, nutrition has major health effects. And you can see here a number of different foods that have been associated, in this case, with cancer and shown to be protective of cancer. Now, those of you who are educated in this field know what's coming next, which is there's the other half of the graph, which shows that, in fact, they've all also been shown to cause cancer. And so basically what you have is, and there are many issues with this, some of them relate to data reproducibility and study number and things like that, but it also points towards a need for systems approaches, which Ben outlined so beautifully in the last talk, as well as to understand that it's not one food is good or bad for every person under every situation. There's obviously personalization here that we need to dive into. So if you look at healthcare today, 86% of healthcare costs are associated with chronic disease. Chronic disease, of course, is pretty massively influenced by nutritional inputs. And so, in fact, if you look at a study that was done in 2007 in the New England Journal of Medicine, you look at determinants of health in the United States. So this is looking at basically what are the drivers of your health over the course of your life. So about 30% of your lifetime health can be attributed back to your genetics, so the hand that you were dealt. About 60% relates back to behavior and the environment, obviously nutrition being a huge part of that. And it turns out that only about 10% of your lifetime health is attributable back directly to the healthcare system. So that's a pretty striking figure when you consider that we spend roughly $4 trillion a year on the healthcare system, which is roughly a fifth of the economy, and it's been growing at 9% per year over time, which is obviously unsustainable. This leads into all kinds of big issues that we could get into in terms of the economics. There's been an estimate that this increasing cost of healthcare creates about a 3% drag on the U.S. economy, which, if you think about that, if you're moving from 1% decline to 2% growth or 1% growth to 4% growth, is a massive shift in the lifestyles of most people. So it's a huge, huge national issue. So we're spending a ton of money on healthcare. At the same time, there is also a commensurate and very large wellness industry. And what we see emerging is a new area that we call scientific wellness, because the wellness industry suffers from a lack of data in many cases. And so what we see emerging now is essentially the ability to build something called scientific wellness, which has at its base very strong data streams that are coming from wearable devices, from omics data, from all this information that we can now underpin so that we can have a forward-looking approach to trying to optimize health and minimize disease before it happens, in addition to what is happening in the current healthcare system. So along with Lee Hood, we wanted to think about something that we could do that would be positively impactful in this space. And so we announced an aspirational project a couple years ago that we called the 100K Wellness Project. And Lee and I put that out there a little while ago. And basically the idea was to recruit 100,000 individuals and do whole genome sequencing on everybody, and then multiple times per year measure proteomes, metabolomes, microbiomes, clinical chemistries, wearable devices, and so forth, and essentially with a pretty big audacious goal, which was to generate data clouds that were dense enough in the sense that they had a lot of different kinds of information, and dynamic enough, meaning that we could look at people enough over time so that we could identify the early warning signs for the major human diseases, to predict them and prevent them. And so that was sort of the genesis of this idea that we announced. Now, this was a fairly audacious announcement. I remember having this conversation with Lee because, just back of the envelope, you think to yourself, that's between a $1 and $10 billion project. And at the time, we had raised $0. So I thought that was a pretty audacious thing to announce. If you know Lee Hood, right, to many people, the father of personalized medicine, he's very undaunted by these kind of things. And I remember even having this conversation with him of, Lee, maybe we shouldn't call it the 100K wellness project. Maybe, let's say we got 50,000, that would be pretty impressive. But if we say 100,000, it sounds like a failure. And Lee gets sort of exasperated, like, Nathan, it's 100,000. It's okay. So we announced this to the world. Now, what's really exciting is that as we've launched this, what has happened is that we actually think we're going to get a lot bigger than this over time. It's taken a little bit of a different form, and I'm going to explain that in a moment, but we're really excited about the possibilities of doing this. So what we've done over the past 2 years then is to build essentially 2 integrated directions that build towards this area of scientific wellness. So one, which was mentioned, is that Lee and I, along with Clayton Lewis, who's our CEO, spun out a company from ISB called Deriving. And I'll describe that in a moment, just a little bit. But basically, Deriving is a consumer-facing scientific wellness company. We recruited about 5,000 individuals in the first 18 months, and it's growing rapidly. We've got 125 employees now, roughly, at the company. And we've raised about $40 million around it. So that's an element that's growing. And what's really exciting about that is that for people who come into the Deriving program, if they check a box saying that their data can be used anonymously for scientific research, then that data comes back into ISB and essentially allows us to do discovery work towards trying to understand the early warning signs for disease and other elements that are related to wellness. And we're very grateful and gratified that so far over 90% of the people who have come through have donated made their data available anonymously to drive that forward. On the other side, the Institute for Systems Biology this last year has become an affiliate of Providence Health, which is the 3rd largest healthcare system in the country. And Lee Hood has now become their chief science officer. So this is a 50-hospital system in 7 states. Providence is actually putting 2,000 of their own employees through the Deriving program as well. But what this is enabling us to do is to drive initiatives around particular disease states, starting with a major initiative that we'll be launching this year on Alzheimer's disease, where Providence has already committed $10 million to that. But basically, this is going to let us drive forward many different scenarios so we can look at the impact of scientific wellness approaches in many different scenarios as well. Okay. So those are the 2 integrated directions that we are taking, and this is driving now the creation of many of these dense dynamic personal data clouds, and we expect to be well over 10,000 by the end of this year and to grow rapidly from there. So all of this was essentially initiated by a project that we undertook in 2014, which was the basis for everything, which we called the Pioneer 100 project. So when we had announced our ambitions towards the 100K project, what we wanted to do was take a finite size project, which was to get 100 people, and it ended up being 108 people because we kept having individuals we really wanted to be in the study kept asking to get in, so it crept up just a little bit. But we have 108 people, mostly in Seattle, but a few others scattered around the country, and basically took them through this process of getting their whole genome sequenced and all the other assays. So we did whole genome sequencing on everyone, so that's millions of differences just in terms of SNPs between individuals. We do clinical lab tests, so about 150 different clinical chemistries, about 700 to 1,000 metabolites, depending on how much came out of the mass spec on each person, about 400 different proteins. Gut microbiome, we also did all of these, by the way, 3 times per year. So we also have gut microbiome. And the self-tracking, so this is mostly Fitbits and things like that. And there were a few other ancillary measurements out of saliva and urine and things like that. But basically, this was a 9-month study, measurements every 3 months to take them through a process. And then what we also did was to take all that information and distill it for them to find actionable information. So this isn't about trying to predict what's going to happen to you in the far future. It's about trying to understand what's happening in your body right now that you can do something with. And so we paired them all with a wellness coach to walk them through that. What this is doing is this is enabling us to build a database of actionable possibilities. What I'll show today is just a few results from the first 100, but as I said, we already have thousands of people that we've done the same thing now on via the Aravail program that came out of this. So this is creating what we call dense dynamic personal data clouds for each person. As I mentioned, we paired them with wellness coaches. For the Pioneer 100, Sandy Kaplan was the wellness coach. I'll say by far the most popular person in the study. People really grew to love Sandy, and also Craig Keebler, who was a terrific physician that oversaw whether there was medically actionable information for people. So what do we see? So just in the very beginnings of this, we can look, and I'll just show, it's a very brief time today, so I'm just going to show a teeny bit of this. But basically, the first thing we can look at are just markers, and this shows essentially the improvement in 6 months. So between the 3-month time point and the 9-month time point for markers related to a variety of different conditions. So cardiovascular, which changes rather slowly, so we saw a 6% increase there. Diabetes improved by 33%. I'll show that in more detail on the next slide. Inflammation by 12% and nutrition by 21%. In terms of clinical labs for diabetes, so these are your standard clinical labs, just the integration of the data with the health coaching, we saw improvement in hemoglobin A1C by 38%, fasting glucose by 19%, and insulin resistance actually over 50% improvement in the number of people who were out of range based on standard clinical measures. All right, so that's just with one data type. We also do things like monitor for various different toxicities. This one was interesting. We had 2 people actually that had this exact problem, or 3 in total, that had very elevated mercury levels. In this case, there were 2 people, and I can actually say them publicly because they've come out and blogged about it quite a lot. Jeff Bland, if you know him, is one of them, and Ed Lazowska, who's the Bill Gates Chair of Computer Science, is the other. But they both had really elevated mercury levels, and it turns out have a great love for tuna sushi. So they were eating tuna sushi a lot. They substituted for salmon sushi. Basically what we saw was in 3 months, it halved the level of mercury, and the next raw, they were totally normalized. The other person is a champion triathlete who is in extremely good shape and considered himself pretty perfect on most measures when he came in. Turned out he also had very high levels of mercury in his blood. Turned out because of his fillings. And his dentist had been telling him to change them for years. He never would, but as soon as he got the blood measurement, it just showed that they were in fact leaching into his blood and had been for quite a long time. So he swapped them out, and that normalized. So simple things like that that you can see with the data. Go to one that's just a slightly more complicated. So here we can look at wellness to disease transition. So we had another person, he was suffering from arthritis-like symptoms in his knees and ankles that had limited him so he couldn't hike with his family anymore, which is something that he really loved to do. And he had concierge medicine on both coasts, and they'd given him some drugs, but they hadn't really worked. But basically, when he came into our study, we did this sort of 360-degree view, and in essence, what happened was something very simple popped out, which was that the ferritin levels in his blood were very high, which is an indicator for potentially hemochromatosis. We looked in his genome. He was homozygous for those genes. And for him, basically, it turns out one of the lesser-known side effects, but possible elements of hemochromatosis, that iron can catalyze cartilage breakdown in the body and can lead to these symptoms. So anyway, we referred him back to the healthcare system. They diagnosed him, in fact, with hemochromatosis, and he got the treatment for that. Now, in this audience, people might know that. This was new to me. But the treatment for hemochromatosis is actually dead simple. You donate blood once a month. So he donates blood once a month, iron levels, the ferritin levels in his blood normalized, and it's N of 1, but those symptoms went away. And so he's back to his active self. And so what was interesting there is that because he had knowledge, he was empowered to make a very simple decision that altered the trajectory of a disease in his future. And so I think it's just a simple case. Diabetes is another case that's very clear, where we already know how to eliminate, by behavior change, disease trajectories in the future. And there's many cases like this that you can dive into and see. And just to show some numbers on that, here are ferritin levels for individuals who have either 0 copies of the gene one copy of the gene or 2 copies of the gene. Now keep in mind the number of individuals, so this is only 2 individuals, but you can see that the trend is the direction that one might expect. And the 2 individuals is important because you have those really elevated levels from the person I just talked to you about. The other person does not have elevated levels. It's a woman, and she's premenopause. So the other interesting thing about this is that you can just let her know, okay, you have the genes that encode for, or that give increased risk for hemochromatosis. Post-menopause, it's possible it will manifest, but if it does, there's just donate blood, it takes care of it. So again, you just, you know about it, there's an action, you can just eliminate it and give peace of mind. And in fact, in the individual that did the treatments, let's see here. Okay, so I want to spend just a couple minutes, because we've just got a few minutes here, but I also wanted to just point a little bit towards where we're going in the future. So the stuff you just heard, I think, was useful. We had some really nice health outcomes for people, but they are necessarily based on what we're already doing today, right? And that's all by regulation. You can't do anything that's different from what's already approved anyway. So that's what you've seen. But now what we can do is just to give you just a teeny glimpse of where we're going with the data. So this figure here is the beginnings of this, but essentially this circle diagram just shows all of the correlations that cross different data types that withstand multiple hypothesis corrections. So these are all statistically significant in the data. So it tells you how the microbiome is related to metabolites that you observe in your blood. It tells you how metabolites in your blood are related to certain lifestyle variables. It tells you how certain genetic traits, in this case predisposition for diseases, are related to your clinical labs and so forth. And one thing I just think is worth contemplating, this is the first time that all of these measurements have been done on the same cohort of people. So even though this is a fairly straightforward analysis to begin with, it's the first time that we've been able to look at that. And that's really meaningful, because that is the tip of an enormous iceberg that we as scientists, as researchers, as people are just barely starting to understand and to even have measurements about. This really isn't happy.
It's not changing. He's coming up.
Huh. I give this talk a lot. I don't. My first technical issues. Really. I don't know what. Yeah. Anyway, so I'll just tell you. We'll see if we can see any of this or not. So one of the things you can do with this big network is you can break it down into modules. So there's algorithms that we can do this that I won't go into in the interest of time. But essentially we end up with functional modules of what's the highest density connections. And what you can start to do is to then understand and see, here are key players that we know what their relationship is to health, but it starts filling in all this other information that maybe we didn't know about what's associated. And in the interest of time, I'm just going to give one example that I think is quite striking. So in one of these cases, what we can do is we can take your genetic risk and use the genetic risk itself as a phenotype. Okay? And I'm going to try to explain that briefly, because what we're going to do is we're going to convert genetic risk into dynamic measures that are that show how that risk is manifesting in the body, and also how then we can intervene on it, including intervening by nutrition and so forth. And so what's really fascinating here is that we can take information. So for example, one of the risk scores we have, and this was really a paradigm shift for me when we first came up with this idea of having the 100,000 people. One of the rationales for that was just we had to have enough people come through who would actually get the disease, so there'd be sort of an end there that we could study. What I realized as we were going through this process, though, was that in fact we could do a lot with fewer numbers by not focusing on people who have the disease, but understanding and doing studies related to everyone's risk for disease. And so this is a subtle point. So one of the things here in these In this information, you'll see, so inflammatory bowel disease, is this going to respond? Okay. Inflammatory bowel disease, this is a score that we have for the genetic risk for inflammatory bowel disease. And you can see that that genetic risk is related, in this case, to the levels of cystine in the blood. Okay? So when we dive in on that, it turns out to be quite interesting. So we find this association, this negative association, the higher someone's risk, genetic risk score prediction for IBD, the lower their cystine is on average. And then we go into the literature and we find that there's support for this in the actual disease scenario as well. So ulcerative colitis activity, turns out it also has this negative relationship with, in this case, with cysteine, which cystine is the reduced dimer form of cysteine. And there's also a case control study with cystine itself that shows that it's one of the top candidate biomarkers for IBD when you do case control studies. Now that's very interesting because remember, we find this same relationship, but we find it in a population in which nobody has IBD, just stratifying by risk. So it tells you at least two things that are very interesting. One is that we could potentially do a correction factor for essentially any case control study in the world. Okay, and why do I say that? So if you do what we and everyone else would do, you get your cases, you get your controls, and you right? And so you have the people that get the disease and those that don't, and you do it, and they get it at the same age, they're matched for age, for sex, for smoking status, whatever. It stands to reason that those that got the disease were at higher genetic risk for that disease than those who didn't. And what this tells you is about all the different changes that you would expect to be there as a function of that genetic risk. So it disambiguates differences that were there in the long lead up of life that led to, potentially led to that disease as opposed to the manifestation of that disease. So this might say, all right, you see cystine popping up as, hey, this is a really good biomarker for IBD versus not, it's related to the disease. But again, we find that difference in a population of people aged 20s all the way to 88 plus with no phenotype. So that's one. The second that's even more enticing is that this might be something that lets us see the manifestation of the genetic risk. In other words, here's the hypothesis, has to be tested, but here's the hypothesis that comes out of it. Cystine sits upstream biochemically of glutathione pathways that combat oxidative stress. So when you see that, what that tells you is that IBD is triggered by oxidative stress. So that lower amount of cystine over the decades of your life means that you have a lower ability to combat oxidative stresses, so it just means that you have a higher probability for transitioning to IBD. Right? So that gives you a mechanism potentially for how this is related to that disease. So what it does then is it transitions you from saying this person's at high genetic risk to here's a potential way that you can mitigate that genetic risk by understanding its manifestation. Now that particular example may or may not hold up over time, but I think the general principle essentially has to be true, which is that gene variants don't mean anything in a test tube, they mean something in the body, which means that they manifest in the body, and by using this dense dynamic personal data clouds, the broader we can go, the more likely it is we can see the ripple effects of what that genetic risk is doing, how it's manifesting, and once you understand how it's manifesting in these dynamic variables and so forth, then you can dive in and modify it without doing anything to the genes, right? But just modify it by diet, nutrition, lifestyle, whatever, pharmaceuticals, and so forth. And so that's really the paradigm that I think is important here. That's frozen again. So anyway, I will just end here then. Let me thank a few people really quickly. First, Lee Hood, who's been just an amazing mentor and partner over many years, and also Andrew Magus, great former bioinformaticist in my group. He's now at Airvale, but he basically did almost all the analytics here, along with John Earls, current student in my group. And so I'll just thank them. Many other people I should thank, but I thanked a few of them along the way. Thanks.